welcome everyone. My name is Gissette Reyes-Soffer, and I'm going to be moderating the next session, which is the session on apolipoproteins, lipoproteins, and lipid metabolism. I'm really excited to be part of this session and co-chairing with Jordan Miller. The session is going to go as follows. I'm going to speak a little bit about the understanding that we have about lipoprotein little a and its cardiovascular disease. And I'm really going to be concentrating on whether we're there yet, of knowing what that role is and how it impacts cardiovascular disease, not only from a clinical perspective, but also from a basic science perspective, and sort of highlighting what the AHA scientific statement finds as a need in this field. And then I'm also really excited because during the session, I get to listen to 5 up-and-coming scientists who have amazing protocols in science that are going to be presented. So congratulations to those presenters, and I look forward to hearing your talks.
My name is Ainara G Cabodevilla. I am a postdoctoral fellow at Ira Goldberg's lab in NYU Grossman School of Medicine. I am very excited to be here to share some of my recent results studying the interaction of the endothelium with ApoB carrying lipoproteins. We recently published a paper in which we showed that endothelial cells are able to take up nascent chylomicrons via the SRB1 receptor, and we're now interested in understanding what the consequences of that interaction and that uptake are in the context of cardiovascular disease and atherosclerosis.
My name is Diego Lucero. I'm a research fellow at Alan Remail's lab at NHLBI. And I'm presenting about CRISPR-Cas screen that we generated to discover new genes related to LDL endocytosis in liver cells. We previously presented in a previous ATVB conference, and also we recently published a paper about some genes that when knocked down, they decrease the LDL uptake. But this time I'm going to show some results that we have that actually some genes that when inhibited or knocked out, they increased LDL uptake, and that could be promising a potential target for future therapeutics.
My name is Hayley Powers. I am a 4th-year graduate student at the Medical College of Wisconsin in Dr. Daisy Sahu's lab, and I will be talking at Vascular Discovery about how our body gets rid of plasma cholesterol. The most common way is through the reverse cholesterol transport pathway, in which high-density lipoproteins deposit cholesterol into the liver, and cholesterol can then be excreted as bile. The main mediator of the movement of cholesterol from HDL into hepatocytes is HDL's receptor, scavenger receptor B1. My talk will be focusing on resolving a high resolution structure of the HDL receptor SRB1 and some of the steps that I've gone through to get there.
My name is Vishal Kothari, and I'm an acting instructor in the Kind Bonfriend lab at the University of Washington. Our lab mainly focusing on the understanding the molecular mechanism of cardiovascular disease in diabetic condition. And our lab previously shown that the triglyceride lipoprotein and its remnant particles, how it can impact the cardiovascular condition in mouse and human studies. This project mainly focusing on the studying the role of the cholesterol ester transferase protein, mainly talked as a CETP, and how it can remodel the TRL and its lipoproteins and affecting the HDL and equivalent functions in diabetic condition, particularly type 1 diabetic condition.
My name is Kendall Burks. I'm an MD, PhD candidate at Washington University in St. Louis School of Medicine. I'm doing my thesis work in the lab of Nathan Stitzel. And I'm really excited to be here and interested in addressing the question of how ANGPTL3 deficiency leads to LDL cholesterol lowering. Specifically, we observe this in an LDL receptor independent manner. And so members of this audience are probably familiar with ANGPTL3 as the target of inhibitors that lower triglycerides and cholesterol. And so we're really interested in teasing apart how LDL specifically is being lowered, with implications for elucidating an LDL receptor independent pathway.
These talks sound terrific. I welcome you to ATBB. I welcome your science. I welcome your enthusiasm. It's amazing to be back in person, and I'm really looking forward to discussing this data with you and to celebrating all your accomplishments, and congratulations on all these wonderful abstracts.